Hello everybody. We're going to talk about your pancreas and your liver in this video. Pancreas is short and sweet. You guys know that your pancreas is nestled right here underneath your stomach and it has a bile duct running down it. That is your pancreatic duct. Right, there you go. The rest of this video we're going to be talking about your liver and this kind of good stuff. So we are switching models to the liver that pops out of our lovely Leslie that you can see fits right there, and your hepatocyte models for this one. Right? So the liver proper. Right? This is your liver. Right? Normally it's a little bit more straight in your body, just like that. Right? Your liver is divided up into lobes. You can donate these lobes if you have somebody in need of a liver and that kind of thing. Your liver is remarkably resistant and can grow back to some degree, right? So we're going to talk about your liver lobes. The two most prevalent ones that are right in your face. This biggest one right here is on the right. This is the right lobe of the liver. So the right lobe. The pointy one that's on the left, guess what? That's the left lobe of the liver. The next two lobes, we have to flip your liver upside down to see. You have a ligament running down the liver right here that separates right and left. That ligament is called the falciform ligament. And now we're going to flip your liver over. Bear in mind, your textbook may get weird with this, because some textbooks flip the liver like I just did, right? That's how normally I would flip it if I was studying with this model in lab. Some of your textbooks, though, not only flip it like this, but flip it upside down as well. Just be aware of what you're studying and know what it looks like. Right? So if this is how I can get the liver to stay, it's going to stay that way. There are two extra lobes. You have this one, no, don't move. You have this one here that is like a little lobe next to the inferior vena cava. That little lobe next to the inferior vena cava looks like a little tail. That is called the caudate lobe. In most biology, when we refer to something as caudate, it looks like a tail. So for those of you once again going into veterinary medicine or zoology, there you go. Caudal is tail. So this is your caudate lobe. Right? The next lobe on the bottom of your liver is this square-looking one here, right? It's a square, which can also be called a quadrangle, which is why colleges have a quad that you can hang out on. That is called the quadrate lobe. Quad is in four, because it's four-sided, it's a square. Quadrate lobe. So you have the big right lobe, pointier left lobe, and then when we flip it over, square quadrate lobe, and the tail-like caudal lobe, or caudate lobe, caudate lobe. Right. I've mentioned the falciform ligament being the ligament here that separates right and left. This white ligament on the bottom of this model here is called the round ligament. Right? You guys can see right here the inferior vena cava that runs next to the liver. So inferior vena cava. Right? This offshoot vein right here that is dumping all of the used blood back into this inferior vena cava, this is the hepatic vein. This is the hepatic vein. The artery that feeds the liver is this artery right here called the hepatic artery. Right? This is where things get complicated. See this lavender looking blood vessel here, and it's the only thing that's lavender colored on the Leslie model, and on most other models it's also blue like the veins. This is the hepatic portal vein here. The hepatic portal vein is bringing blood that from the intestines that just absorbed nutrients to the liver so your liver can do its job as a filter and poison control center. So the blood that is in your hepatic portal vein is going from intestines to the liver to be filtered. And then after your liver filters it, it will then go into the hepatic vein here and dumped back into the inferior vena cava. But it's not clean yet, so it has to be filtered. That's why it's in the hepatic portal vein. Right? Also, on the underside of this liver, you can see the gallbladder right here that stores extra bile that your liver makes, and the cystic duct that connects to the gallbladder. So you have cystic duct, gallbladder. Right? These are also the hepatic ducts that are dumping into the common bile duct here. Right? Everything else that you need to know about the liver gets up close and personal with the liver, which is why we are going to start using our lovely liver cell model. So your liver is divided into lobes, or little lobes. They are called lobules. This is a unique sort of model with a unique sort of system from what you guys need to know. So it's kind of fun and easy to tell that these are liver lobules and that this is your liver. First of all, it's the only digestive system model that is kind of purplish in color. So if you're one of my color people, there you go for that one. These individual little cells within your liver are all called hepatocytes, because hepatic means liver. You guys see where this is going. Hepatocyte, liver cell. Right? So each of these little lobules that your liver creates, so each of these lobules here is hexagon shaped. Notice that it's not perfectly round. It has corners. Like, these aren't round. They have 6 sides. They have corners. Each little lobule is not round. It is stop sign shaped, which also makes this kind of unique. So if you see stop sign shaped lobules on your final exam or any models, this is your liver, right? So these are the lobules, and down the middle of each of these lobules is something called the central vein. The individual cells within these lobules are what's doing all the filtering inside your liver. So all of these tubes that lead to, that are going down the middle of the lobules that are doing the filtering, all lead to this central vein here. And these little tubes that are going through the lobules, these spaces here, these are called sinusoids. But what liquid is being filtered, you may ask, that's being dumped into this central vein here? Notice how the corners of each of these lobules right here, right? Each of the corners of these little stop sign hexagons have 3 little vessels there. That is called the portal triad. Within the portal triad, you have a portal arteriole, so a little artery that's running through to feed the cells in the lobule, a portal venule, which is a small vein that is taking used blood, right, from the cells, and a bile duct, which is where waste byproduct is being dumped so that it can go back into the digestive system and then be removed from your body with fecal matter. That's what bile does, or one of the jobs of bile, I should say. Right? So you have these lobules that are stop signed or hexagon shaped, and in each corner of your hexagon you have a portal triad here. So you have a portal triad in each corner of these hexagons. This vein here has all the liquid from the hepatic portal vein. So everything that your body absorbed to try and gain nutrients from is going through these little venules here, and then is leaving the venules and filtering through these sinusoids, and the individual cells or hepatocytes within the lobules do their job and filter the bad things out of the liquid, and can let it all continue. They filter the stuff that your body does not need, or they do things like a lot of enzymes in here will also convert things like hydrogen peroxide back into gas and water. If you'd actually like to see that happen, go to the grocery store, get some chicken livers for like a couple of bucks. And if you put hydrogen peroxide, even if the liver is dead, like what you buy at a grocery store, if you put hydrogen peroxide on it, it will start to bubble. That is an experiment you can do in your own house if you really want to, right? So you have yeah, it will convert things because you don't need to drink hydrogen peroxide. And as everything filters through from these venules through these sinusoids to the central vein, it's going to be filtered. So once again, stuff is going to come out of these little venules through the sinusoids and be filtered and then be clean when it hits the central vein. The only other thing you guys need to know about, right? So the only other thing you guys need to know about is on some of these models, you can see that within these sinusoids, you see some of these amoeba-like white blood cells that are here. These are a specific type of macrophage that patrol in your liver to make sure they eat any bacteria that you might have accidentally absorbed from food. And these little macrophages are either called hepatic macrophages or, like your lab list calls them, Kupffer cells. So those are the type of white blood cells that help your liver. I hope that made sense to you guys, and I hope you have a wonderful evening. Have a good night.